It's easy to imagine that bodies simply wear out like old cars, with parts slowly rusting and failing over time. But 2 recent articles look at cellular decay and find something much more deliberate at work. In Quanta Magazine, Ingrid Wickelgren asks if mammalian aging is actually a tightly coordinated schedule running inside the cells, rather than random physical damage. Meanwhile, a piece from Smithsonian Magazine looks to the ocean to see how octopuses keep their long-lived brain cells healthy. They rely on a rare genetic trick to force perfect accuracy when building proteins. Putting these together creates a striking picture of how biology handles time, managing the physical limits of a living body in completely different ways. Writing just last week, Ingrid Wickelgren profiles a cell biologist who thinks science has aging all wrong. People usually picture getting older as a slow breakdown of parts, like a factory with rusting equipment. But listen to how Wickelgren uses a massive experiment on 21 million mouse cells to flip that script. The data points to a startling alternative. Growing old doesn't happen by accident. It might be a strict, orderly program, kicking off before a person even hits 30.
Why aging may be a program, not a breakdown. Introduction. In some ways, we know aging when we see it, from the graying of hair to the wrinkling of skin to declines in motor, sensory, and cognitive capacities. Yet the underlying biology of aging remains a matter of uncertainty and debate. Many lines of research align with the theory that aging is a direct result of decay, the inevitable degradation of molecules, including proteins or DNA, organelles, cells, or whole organs, from external assault or inexorable breakdown. When the body's repair mechanisms fail to keep pace with these changes, like a factory with deteriorating equipment and too few mechanics, it manifests as the known signs of aging, and eventually, death. The idea makes a lot of sense, but according to the cell biologist Junwei Kao, it's inaccurate. Far from a random but linear process of wear and tear, he argues, aging is a stepwise, programmed, orderly affair. Quote, "The destruction of the system is programmed at a very early stage," said Kao, who heads the Laboratory of Single-Cell Genomics and Population Dynamics at Rockefeller University. Using technology that offers a system-wide view of the aging process in mice, Kao has outlined discrete stages of aging, akin to those of embryonic development, that are defined by changes in molecular signals and specific cell populations. In humans, the process likely begins before age 30. Kao's interest in aging began in high school in Hebei, China, when he became acutely aware that his grandparents and parents were not going to live forever. While many teenagers awakening to mortality might turn to poetry or self-destructive behavior, Kao turned to science.
Finding a way to slow aging became his lifelong goal, and it's why he chose biology as his major at Peking University in Beijing. Initially, Kao assumed that the deterioration of particular proteins and protein networks was responsible for aging, in line with the prevailing model. But then, after college, when he was working in a lab trying to identify those proteins, he realized that a daunting number of them were associated with aging, and that their effects depended on the type of cell in which they were operating. It was a picture both more complex and more organized than he had thought. Kao decided he needed data, lots of it, on thousands of molecular changes across hundreds of cell types. As a graduate student, he developed a high-throughput technology that could quantify these dynamics in embryonic development. When he started his own lab at Rockefeller in 2020, he put this technology to work on aging. In one series of experiments, Kao and his team processed 21 million cells, sampled from 14 tissues or organs in about 50 male and female mice at 5 life stages, and built a dataset of gene expression for each cell. It's extremely large-scale data. You know which organ it's from and which age it's from, and you also know extensive molecular information. Each stage was marked by a dramatic decline in or expansion of specific cell types. 2 of his landmark papers, published in 2025 and 2026 in Science, point to a radical redistribution of the cells that make up the body as mammals age, and describe some of the epigenomic instructions that guide this process.
There are molecular changes, and maybe some other changes in aging, Kao said, but they all converge in the remodeling of the cell society. Quanta spoke with Cal about evidence for the programmed theory of aging, what happens at each stage, and why aging mammals are like trees shedding leaves. The interview has been condensed and edited for clarity. You began studying aging as a college student. How has your approach to the topic evolved? Back in college, I thought the problem of aging that needed solving was the lack of a drug. I thought that we were going to develop some magic drug or chemical that we could use to increase the lifespan of animals. So I joined a lab working on computational drug design, trying to design peptides that target molecules related to aging. But the major challenge was that we didn't know what molecules to target. After I graduated, I moved to the US and worked at the Jackson Laboratory, a Maine-based biomedical research profit, as a research assistant studying molecular pathways associated with aging in mice. I thought that by studying pathways, we could identify a drug target, and from there, develop drugs to increase lifespan. But after a few years, I learned that aging cannot be explained by a single pathway or target. It involves many different pathways, and these pathways have very different effects on different cell types in the body. I wanted to understand how aging progresses on a molecular level across hundreds to thousands of cell types in different organs. How did you do that? A major challenge is that aging involves changes across many different levels: molecules, organelles, cells, organs, and the whole body.
To measure changes at all these levels, we needed technology that was both high throughput, which means you can use it to scan not just one cell or cell type, but millions of cells across an entire organism, and high resolution, so you can see very detailed changes at the cellular and molecular levels. I applied to graduate school, where I developed tools that could scan the entire mammalian organism, from the brain to the kidneys to the lungs, while detailing tens of thousands of gene expression changes within single cells across hundreds of different cell types. When I got to Rockefeller, we used this technology to understand the dynamics of the whole system in aging. Can you describe those experiments and what you learned from them? In one set of studies, we extracted more than 20 million cells from various organs from mice of different ages, 3, 6, 12, 16, and 23 months, roughly equivalent to 20, 30, 50, 60, and 75 years in humans. We analyzed the expression of 20,000 genes per cell and used this information to define the cell types. Then we tracked their population dynamics. We found that not every cell type gets changed in aging. We identified 536 main cell types and 1,828 subtypes. Only about 1/4 of these subtypes show a strong shift in aging. Others remain stable across the lifespan. It is surprising to find that changes in aging are not universal across all the cells, that there are specific cell populations that are more vulnerable. When do these population changes occur? We found that aging can be separated into distinct time windows. In each window, specific groups of cells show coordinated dynamics. In the early phase, for example, we see that some cell types are rapidly depleted.
This is followed by another phase in which other cells are greatly expanded. What types of cells are affected at each stage? In the first stage, 3 to 6 months in a mouse, about 20 to 30 years in a human, there is a loss of certain fat and muscle cells, and of 2 immature cell types in the brain that have the capacity to regenerate different types of brain tissue. Between 6 and 12 months in a mouse, equivalent to a person in their 30s and 40s, we see dramatic depletion of cells needed to maintain the body's tissues. These include tenocytes, the primary component of tendons, the cells that wrap around blood vessels and stabilize the circulatory system, the smooth muscle cells of the colon, and kidney epithelial cells, which filter toxins from the blood. Also in decline are some immune cells that protect specific tissues, such as the intestine. At around 12 months in mice, roughly 40 to 45 years in humans, there is a shift from cell depletion to cell expansion. The first expansion wave is dominated by immune cells, but also includes select cells in the lungs, kidneys, and other organs whose properties have changed as a result of stress or inflammation. At around 16 months in mice, late 50s and beyond in humans, specialized aging-associated immune cells expand. When these selfish, uncontrolled cells emerge, they will eventually proliferate and destroy the system. In the meantime, they may contribute to the increased risk with age of inflammatory conditions, such as heart disease, arthritis, cancer, and chronic respiratory illnesses.
Is there evidence that these stages also occur in humans? Yes. Researchers have observed a phenomenon called abrupt aging in middle-aged humans that is consistent with the cell population dynamics we saw.
When researchers analyze protein signatures in human blood, they see a significant change between the mid-40s and late 50s. People also tend to report an abrupt decline in function in middle age, which is also when there is a rise in susceptibility to age-related diseases, such as cancer and some neurodegenerative diseases. What do your findings tell us about the nature of aging? Previously, people saw aging as a linear accumulation of damage to molecules, such as proteins and DNA. But we found that aging is not a linear process. It's more like a developmental process, in which there are distinct stages that involve coordinated changes in specific cell types across different organs. Our claim is that aging is not so much molecular damage as a remodeling of the entire cell society. What controls this cellular remodeling in aging, if anything? Our technology allows us to examine the genomic program that governs the function of each cell, which regions of the genome are active and which are silent. If it's random molecular damage, we might see random changes across the genome. But we always see the same regions that are open, active, or closed, silent, at each stage. We identified 280,000 genomic regions that are reproducibly open or closed during the aging process in specific cell types. So the body has a program for aging? Yes. The abrupt changes in mammalian aging, along with the coordinated cellular dynamics at each stage, suggest there are upstream signals that control aging. It's like a tree in autumn. Its leaves fall not in a linear way, but in just 2 weeks during the transition between summer and autumn. The changes in daylight trigger the release of a signal, and then the system gets changed.
In mammalian aging, the abrupt changes show there is some kind of signaling that drives the system. What type of signaling? We have found both internal molecular programs, proteins that control gene expression, and external, secreted molecules called cytokines that drive the cellular changes. What does your work tell us about how to slow or stop aging? We have identified some rare vulnerable cell types that could be targets for anti-aging interventions, and we have found the molecular code that drives the aging process, so we can develop some approaches to reprogram that code. Crucially, we have shown that the regenerative capacity and robustness of the system decrease before middle age. So if we want to rescue aging, we should start early.
Ingrid Wickelgren leaves you looking at a ticking biological clock. The idea that aging isn't just a rusty factory breaking down, but a highly organized, scheduled shutdown that kicks in before age 30 changes how you view a gray hair or a stiff knee. It feels almost fatalistic, a genetic script no mammal can escape. Turning to the next piece, that sense of biological inevitability vanishes. Looking at shallow water octopuses, the writer finds a completely different approach to cellular maintenance. Where Wickelgren focuses on the scheduled degradation of proteins and cells in mammals, this second story looks at a genetic trick designed to prevent errors in the first place. Notice how the two pieces approach the same basic biological problem from opposite ends. Mammals seem programmed to eventually shut down their cellular factories, but the California two-spot octopus evolved a literal gap in its RNA that forces its cells to build proteins with extreme accuracy. Read alongside Wickelgren, the octopus's mutation reads as a brilliant evolutionary workaround to the very protein breakdown that drives mammalian aging.
Why are some octopuses so smart? The answer might lie in a never-before-seen mutation that helps them accurately build proteins. Scientists discovered a strange feature in certain octopuses' ribosomal RNA, molecules that create a 3D scaffold for cellular protein factories. It was found only in shallow water creatures that have expanded nervous systems and can do complex behaviors. Octopuses are incredibly clever creatures. They can open jars, solve mazes, and even use tools. One species, the common blanket octopus, wields venomous tentacles ripped from the Portuguese man-o-war as weapons. Now, researchers have discovered a mysterious mutation in some octopuses that might explain their intelligence. A study published in the August 17 issue of the journal Current Biology reveals that the 8-limbed creatures can produce proteins with extreme accuracy, thanks to a variation never seen in any other animal. Although there is no direct evidence that the adaptation is linked to expanded octopus brain power, only a lineage of creatures with enlarged nervous systems and that can carry out complex behaviors appears to have the mutation. Scientists made this discovery by accident. About 5 years ago, study co-author Richard Hahn, then a graduate student at Harvard Medical School, was examining molecules called ribosomal RNA, or rRNA, in tissues from the California two-spot octopus. The molecules create a 3D scaffold for ribosomes, the cell's protein factories. Many sequences of rRNA remain pretty much the same across all known animals, but Hahn noticed something unusual in those from the octopus. An unexpected gap that broke what's usually one rRNA fragment in other creatures into two.
We figured we were bad at extracting RNA and simply had made a mistake, says study co-author Nicholas Bologno, a molecular biologist at Harvard, to Sarah Reardon at Science. Further tests, however, confirmed that something else was going on. Inserting the same break in the ribosomes of Escherichia coli bacteria made the engineered cells produce proteins with about twice their usual accuracy. To examine when the strange rRNA feature evolved, the team compared two groups of octopuses that diverged more than 100 million years ago. Inserates, shallow water octopuses with developed nervous systems that support complex behaviors, and cerates, deep-sea creatures with simpler nervous systems adapted for slow swimming and passive feeding. The rRNA break was present in all 5 examined inserate species, the team found, but a sample from a cerate, specifically a Dumbo octopus, lacked the gap. Squids, which diverged from octopuses about 300 million years ago, also didn't have it. Fun fact: self-editing. Cephalopods, an animal group that includes octopuses, squids, cuttlefish, and nautiluses, are masters of editing their own RNA, molecules that carry instructions from DNA to help build proteins. They do it far more often than other creatures do. In a study published in 2023, researchers reported that octopuses heavily edit RNA in their brains to brave frigid water. The findings hint that the rRNA adaptation might be connected to the evolution of the shallow water octopus's large nervous systems. Their brains, which are spread throughout their bodies, had to expand quickly as they learned to keep up with predators and increased competition in this environment. Nerve cells, or neurons, are long-lived, study co-author Rishav Mitra tells Scientific American's Cody Cottier, which means protein misfolding is particularly bad for them. By preventing that, the RNA break might help these neurons to work well, he adds. The major surprise is that the ribosome, which is highly conserved across life, can actually undergo evolutionary changes that impact function and may even contribute to new innovations, study co-author Amy Lee, a cell biologist at Harvard, says in a statement. Joshua Rosenthal, a molecular biologist at the Marine Biological Laboratory who wasn't involved in the work, calls the discovery super interesting, although he notes that more research is needed to prove whether the RNA change drove the evolution of sophisticated brains and behaviors. We're just getting to the beginning of genetics with these organisms, he tells Science. The study authors suspect their findings may lead to potential therapies for neurodegenerative diseases like Alzheimer's disease and Parkinson's disease that involve misfolded proteins in the brain. Lee tells Scientific American that she hopes that it will be possible to design drugs that copy the octopus mutation for accurate protein synthesis. If we use nature as a guide to understand how that happens naturally, she says, then we can probably find ways to put it into human cells.
A mammalian cell follows a quiet schedule, slowly turning down the lights as the years pass. Deep underwater, an octopus neuron uses a genetic loophole to keep its proteins perfectly shaped and pristine. What lingers after hearing both stories is a new way to look at physical decline. Aging suddenly looks like a deliberate biological dial instead of random rust. If an octopus can evolve a molecular trick to freeze that dial in place, why did mammals inherit a clock that guarantees they will eventually break down?